Hello friends, welcome to my channel Naved Biochem Classes. Today I request you to subscribe my channel and share it. Okay. Today we will talk about enzymatic method for determination of sequence of amino acid in a peptide chain from C terminal. Okay. So what is all this about? We are talking determination of sequence of amino acid in a peptide chain from C terminal direction of a polypeptide chain by reaction or by use of enzyme carboxypeptidase. Now, what is carboxypeptidase? Carboxypeptidase is an enzyme secreted by pancreas. It is secreted in an inactive form. When it is secreted, it is not active and it is procarboxypeptidase and which get activated in the small intestine to break down proteins into a smaller peptides. Okay. Now, it is a protease enzyme, that is the enzyme that hydrolyzes a peptide at carboxyl end of a peptide or protein. Any acid which enzyme which hydrolyzes the peptide of protein or a peptide bond is called protease. Now, carboxypeptidase that cleaves amino acid containing aromatic or branched hydrocarbons are called carboxypeptidase A, where A stands for aliphatic or aromatic amino acid. Similarly, carboxypeptidase that cleaves basic positively charged amino acid are called carboxypeptidase, that is B for basic. Apart from this carboxypeptidase A and B, you also have metallocarboxypeptidase enzyme which terminates C-terminal glutamate from peptide N-acetyl, L-aspartyl, L-glutamate and is called glutamate carboxypeptidase. Now another one is serine carboxypeptidase that cleaves a C-terminal residue from peptide containing the sequence PROXAA, where PRO is proline and XAA is an amino acid on the C terminus of a peptide, and it is called prolyl carboxypeptidase. Carboxypeptidase enzyme attacks peptide chain only at C terminal end, which contains free carboxylic group, to liberate amino acid. When the terminal amino acid is residue is liberated as free amino acid, carboxypeptidase enzyme attacks new free carboxylic group at the C terminal end of the remaining peptide chain, and this is repeated till all amino acid is liberated. So you can see, for example, if you have a tripeptide as shown in this figure, and when it is subjected to hydrolysis by carboxypeptidase, the first amino acid, that is C amino acid residue from C carboxylic end, is liberated, followed by B and followed by A. Okay, so here is the example. You can see you have a reaction sequence. You have an example of a tripeptide. When it is subjected to carboxypeptidase enzyme, hydrolysis takes place, and you can see the first amino acid is liberated. You can mark it as a C, and the rest is degraded polypeptide chain, which is again, when subjected to hydrolysis it by the carboxypeptidase enzyme, you get two amino acid, that is B followed by A. And here you should know that quantitatively C is greater than B. So the quantity of C is always greater than B followed by A. So if you know the first amino acid, if you identify it, which is liberated from teripeptide, and if you know the quantitative amount of amino acid liberated by enzyme carboxypeptidase, you can find it out that what are the ABC of a tripeptide is, and what is the sequence, that is the amino acid which will have highest quantity will be at the terminal end. Okay, that is R3 amino acid, you can see here this one, followed by the middle one, that is B, that is the that is the that is the R2 one, and last one the R1 one. So you can see the sequence will be, you can see that sequence will be CBA from C terminal end. So this is all about enzymatic hydrolysis of a peptide chain from C terminal end. Obviously, enzyme in use is carboxypeptidase. It can be A carboxypeptidase A, which means A for aromatic or aliphatic. Then you have B, which is for basic. B stands for basic, which cleaves basic amino acid. It is a specific. Apart from that, you have metallo and carboxypeptidase, and you have also serine carboxypeptidase. And depending upon the uh, sequence on which the amino acid is obtained by hydrolysis of it, and also by quantitative, you can determine the sequence. That is, higher quantity is always at the start of the C terminal position of the amino acid, and similarly it is followed by B residue and A residue. So this is all about enzymatic hydrolysis of enzymatic method for determination of sequence of amino acid in peptide chain from C terminal direction. Thank you, and again I could request that please subscribe my channel. Thank you.